and we'll see the same slides that we would have seen had we do a face-to-face, except you'll have you know, your books and notes and stuff like that to look at them, as opposed to if you're in the face-to-face, you wouldn't, okay? And so we'll start talking about cytology now. Cytology is a study of this. Cytology is just a study of the cell. That's all it is, okay? We have a lot of cells. We probably have 1 trillion cells, okay? If you don't know, trillions 10 to the minus 9, a billion's 10 to the minus 6, 1,000th a trillion, a million's 10 to the 6th, a billion's 10 to the 9th, and a trillion would be 10 to the 12th, okay? I try to, you know, play mental gymnastics sometimes. We have 250 million cell types. And you have to remember that in the nucleus is the DNA, essentially the blueprint for each one of those 250 million cell types. We may have a thumb cell, okay, a skin cell that goes on the thumb, but there is the potential in that particular DNA to be a liver cell, okay? So so in your lifetime, especially if you're younger and you go in the healthcare field, you're going to see more and more being able to regrow tissues, okay? Because the potential's there. It's just a matter of reworking the technology, which would be really good for spinal cord injuries. It'd be really good for heart attacks and heart injuries, because the heart heals by fibrosis. It doesn't regenerate tissue, so it just heals by scar. And so if you knock out 25% of the heart, that's 25% heart muscle that heals by scar, and you've lost 25% of the activity of the heart muscle. Same way with the brain. And so being able to regenerate those tissues by using other tissues, taking a biopsy somewhere on your body and programming that to be able to regenerate those tissues would be really good. And so we have a number of different types of tissues and a number of different types of cells. We're going to talk globally, in other words, big picture about a cell, okay? So we're going to talk globally, big picture about this cell. But when we get into tissues next week, that'll be a pretty involved lecture and a pretty involved lab, okay? And so we'll spend a considerable amount of time talking about tissues and talking about individual cells when we get there next week. Cellular structure, there's 3 main structures to a cell. Now we're talking in general about a multicellular organism. We're talking in general about a multicellular organism. We're not talking about a plant cell. We're talking about an animal cell, because there's differences between plant and animal cells. Plant cells have a rigid cell wall and animal cells don't. So we have three basic structures on a cell are, number 1, a plasma membrane, which is the outer boundary, okay? That's the plasma membrane, the outer boundary. And so that would be this membrane around here. That we have the nucleus. This is the nucleus, one of the bigger organelles, okay? And then we have cytoplasm. So we have the plasma membrane and the nucleus. Everything outside of the nucleus and inside of the plasma membrane, the cell membrane, is cytosol, okay? Everything outside of the nucleus and inside the cell membrane, that's not an organelle, okay? That's not an organelle. Mitochondria organelles, endoplasmic reticulum, we're going to talk about all these are organelles, but that's cytosol. And so that is the gel-like fluid content in a cell. And then we can have some inclusions. If you were talking about a plant cell has a water inclusion, because animals are able, as a general rule, to get water when they want and need it, whereas plants are only able to get water when it's available to them. So they store water in their cells in an inclusion. Okay, we may have an inclusion where we've digested a foreign material and not yet extruded it from the cell. The plasma membrane is a fluid mosaic membrane, okay? Fluid mosaic, which means that within a certain limitation, it has flow to it. And if you could see a dynamic schematic of it, you would see that it's sort of flowing, has some flow to it back and forth, We will talk about this in detail, but the plasma membrane has a bilayer here. This is 2 layers, okay? A bilayer here. And this is the phospholipid that we talked about when we talked about lipids, part 2 of the chemistry, phospholipids. It has a glycerol backbone, 2 long-chain fatty acids, and a phosphate group. Well, these little tops here are the phosphate group. There's one on the outside of the cell, on the outside perimeter of the cell, and one on the inside perimeter of the cell. And they're valuable because they are polar. Remember, we are made primarily of water, okay? We have water and we have minerals and a number of other things, but our transport medium, our fluid around the cells, our fluid inside the cells is primarily water. Water is a fluid, water is a polar compound, okay? In order for the water to come up, in order for this fluid to come up, which is bringing hormones, which is bringing glucose, which is bringing oxygen, which is bringing other things to come up and butt against the cell, we need a polar end because polar attracts polar. Polar attracts polar. So we can attract, so if this is the phosphates a negative group, it would attract the positive end of the water molecule, which would be the hydrogen end, partially positive. And so that's the way we're able to get the fluid. We have the bilayer of fatty acids here. Fatty acid, the phosphate group is hydrophilic, which means it likes water. The fatty acids are hydrophobic, which means they don't like water. So this is our one way of our regulatory mechanism of regulating what goes in and out of the cell. What goes in and out of the cell is that biphosphate layer. Let's talk a little bit about what the fluid spaces are. We have intracellular fluid. We have intercellular fluid, which is a fluid inside the cell. We have extracellular fluid, okay? We have extracellular fluid, which is primarily 2 components. The first component is the intervascular component, and the second component is the interstitial component. There is no direct connection between the cells and the blood vessels, no direct connection. All the cells have blood vessels around them. All the cells have blood vessels around them, but there's no direct connection. So for glucose to get to a cell, glucose has to be transported to the cell by way of the vascular space, has to come out of the vascular space into a fluid that's bathing the cell. So the fluid that's bathing the cell is called interstitial fluid, okay? So we have intercellular fluid and extracellular fluid. Extracellular fluid consists of the fluid that's in the vascular space, okay, inside the blood vessels, and the fluid that's outside the vascular space, which is bathing the cells. And so, I mean, we're not talking about long distance. We're talking about, blood vessels are surrounding all the cells. So they're just right here. I mean, they're just right here on the periphery outside, but there is no direct connection. So that material goes out of the blood vessel into this space. And then it's in this space here. So the glucose would come out of there, would come up and butt against the cell, okay? And then would be transported into the cell from the intravascular to the interstitial, then to the intercellular, okay? So that takes a little while sometimes to understand, and it took me a while to understand what that meant, okay? We have the phospholipid bilayer, which we talked about, okay, which were the lollipop-shaped phosphate molecules, which are polar, when they're hydrophilic, okay, attracted to water, intercellular and extracellular fluid. So we need them on this side here because the intercellular fluid is primarily water. The extracellular fluid is primarily water, okay? And then we have the hydrophilic areas, which are the biphospholipid layers. The hydrophilic areas, the biphospholipid layers, okay? If we had some phospholipid in a beaker and we got a eyedropper and we picked up a little bit of it and dropped that into the water, okay, into a beaker of water, it would form a droplet. And the droplet would be with the phosphate lollipop, the round sections pointed outward towards the water, and all the fatty acids were pointed inward. So that's why fat forms droplets. If you have a fat water mixture, like in a lot of dressings, that's why it forms droplets. Lipids Lipids also have sugar groups attached to them. So these things sticking up here are sugar groups, okay? We have cholesterol. These are cholesterol. This is cholesterol. These oval things here are cholesterol. They stabilize the plasma membrane and decrease some of the fluidity, okay? You don't want it completely fluid, okay? So they add stability to it. We have carbohydrate-rich areas, okay? So some of these are lipids out here. Some of these are carbohydrates, and they're responsible for cell-to-cell recognition. We have survey cells or scavenger cells that are going around our body all the time looking for non-self, okay? We have antigens, and we have identifiers on most all our cells in our body, 99.9% of the cells in the body. We have identifiers on them saying they are self. And so everyone in this class has their own identifiers that are identifying themselves. And unless you have an identical twin, there is nobody quite like you, okay? And so the purpose of that is the scavenger cells come around. And if you've been invaded by a foreign bacteria or a virus, or if you have some foreign material inside of you, it will identify it as non-self. Identify it as non-self. If it's small enough, it will try to get rid of it, okay, by a number of mechanisms, and we cover that extensively, anatomy and physiology too. If they can't break it down, then they will try to wall it off, okay, and they will try to wall it off. And so you have, you know, if you have some artificial thing placed in your body, then there's usually a capsule formed around it, okay, because obviously if you have a knee replacement, it's not going to be able to, body's not going to be able to digest that stainless steel. Stainless steel is not very reactive. So let's think of something else. You break off a piece of pencil lead, okay, or graphite in your hand, okay, which I did a long time ago, body walls that off, okay? The body's going to wall that off because that's too big for the white blood cells to take care of, okay? So that's cell-to-cell recognition. We want to be able to recognize self as opposed to non-self. Integral membrane proteins, that are these proteins here, is the proteins here. Some of them go all the way through, some of them don't. Some of them attach, okay, are peripheral proteins and attach on the outside and on the inside, okay? And they extend through the phospholipid layer. Peripheral proteins are loosely attached, okay? So we can have some peripheral proteins that are loosely attached. They're responsible for most of the specialized functions, okay? Transport, that one that had this one up here that had the transport thing, that could very well be a transport protein for water, call it aquaporin, okay? It would regulate how much water, wouldn't be freely available to it, would regulate how much water you want into the cell, okay? They could be receptors for signal transduction, attachments to the cytoskeleton, okay? We have a cell, and the cell has some fluidity and has some movement to it, but it basically needs to have some type of structure. They could be responsible for enzymatic activity. In other words, they could start an enzyme process that's related to the cell, whether it be taking in a particular item, like a thyroid hormone or something like that. I'm just giving that as an example. Intercellular joining or cell-to-cell recognition. So these are some of the functions of the membrane proteins. Membrane transport is selectively permeable. In other words, everything doesn't get in, okay? Now, oxygen goes right through. CO2 goes right through, okay? So oxygen, when it's delivered, it goes right through the membrane, right into the cell, okay? CO2 goes right out of the cell. And there's some other things like that, but otherwise, the plasma membrane is selectively permeable to the material that it's going to admit or it's going to remove. Things move back and forth from the extracellular space to the intercellular space. The extracellular space, one of the components is interstitial fluid. Remember, that's the fluid, that's extracellular fluid that is not inside blood vessels, okay? Extracellular fluid that's not inside blood vessels, interstitial fluid. They are a number of methods of passive transport. What is passive transport? Passive transport does not require energy. In other words, it doesn't require ATP. So I would listen carefully. I would make sure I knew this material, because I can see a short answer question coming, on your exam. Differentiate, the different types of passive transport. And they're not hard, okay? They're not hard. No energy is required. The first one's diffusion. You know about diffusion. Diffusion is the movement of a substance from an area of higher concentration to an area of lower concentration down a concentration gradient till equilibrium is reached. Here's an example. If we were in a face-to-face classroom and we had pretty good seals on the door, I could open up something, like a can of sardines or a can of really obnoxious, men's cologne or something like that in the corner of the room, okay, in the corner. Or I could open up and release some hydrogen sulfide gas, okay, in the far corner of the room. It would not be very long before that hydrogen sulfide gas, just a small amount of that small amount of sardines. We had somebody in our medical school class, one of my friends, who liked to eat sardines after lunch. He would do something at lunch, maybe study, and for the 1 o'clock lecture, he'd come in and he'd pop a can of sardines. Well, you could smell that all over the lecture hall, because sardines have a particular aroma to them. And so that smell, those odor atoms, okay, whatever the odor is, okay, would diffuse throughout the entire room. Doesn't have to go through a membrane. There is no membrane, okay? There is no membrane. It would diffuse throughout the entire room. And eventually it would reach equilibrium, so that if I sampled the air right next to the container, because all that smell would have come out, all that odor would have come out, the odor atoms would have come out, and every place I sampled in the room would have the same number of those men's cologne molecules, obnoxious men's cologne molecules, and that's diffusion. So diffusion is the movement of a substance, okay? It can be a liquid, it can be a gas, it can be a solid, okay? Movement of substance from an area of higher concentration to an area of lower concentration, down a concentration gradient, okay? Because you're going from high to low, until equilibrium is reached. That is diffusion, okay? Particles move from an area of high concentration to an area of lower concentration, okay? So lipid-soluble solutes, very small ones, just go right, if you're lipid-soluble, it goes right through the plasma membrane. And we go, we'll go from high concentration to lower concentration. Facilitated diffusion is sort of a unique thing, okay? You have this is a solute. A solute is just some kind of dissolved thing. It can be so a solute, a particle cannot. It requires a transport protein to get it through, some kind of transport. Okay, but it doesn't require any energy. So you have these particles here, lipid insoluble solutes such as sugars or amino acids. They wouldn't normally go through the plasma membrane. Okay. And so you have this transporter here. It's opened up. So one of the solute, and these are specific for specific solutes. Everybody can't catch a ride. Just this one particular solute, okay, goes into this transport protein. The transport protein closes and undergoes a configurational change, and releases it on the other side, releases it inside the cell. So this is carrier-mediated facilitated diffusion, okay? But it doesn't require energy, which is astounding, because the protein has to undergo a configurational change. So this is facilitated diffusion. Osmosis is a specific type of diffusion that requires two things. Requires water and a semi-permeable membrane, okay? Notice we haven't talked about any kind of membrane before, if we're just talking about simple diffusion. We're just talking about simple diffusion, except for the plasma membrane when we're talking about facilitated diffusion. But the definition of osmosis, there's two definitions, okay? I'll give you the technical definition first. Osmosis is the movement of water through a semi-permeable membrane from an area of high concentration of water to an area of lower concentration of water till equilibrium is reached. Osmosis is the movement of water through a semipermeable membrane from an area of high concentration of water to an area of low concentration of water down a concentration gradient until equilibrium is reached. Now, the other definition of diffusion, of osmosis, okay, water moves from an area, water moves through a semipermeable membrane from an area of low solute concentration to an area of high solute concentration until equilibrium is reached. And solute is just the number of dissolved particles in solution, okay? So if you think about it, the water is moving in to try to dilute the more concentrated whatever vehicle's there, and it's going through a membrane. Okay, so there's two different definitions. You can use either one that you want to, but technically it's the movement of water. Okay, but when we get to the slide, you'll see there's a slide that says water follows the stuff. Okay, so if you got water and in one area and a semipermeable membrane that's semipermeable to water, okay, the water will move from that area because there's less stuff to an area where there's more stuff. Okay. And so the channels for water movement are aquaporin, okay? They're aquaporin. And it's regulated because we can't, obviously the cell is very concentrated. You know, we've already started looking at the cells, very concentrated in comparison to the interstitial fluid. So if the plasma membrane was freely permeable, okay, it's freely permeable to water, water would just rush into the cell and the cell will grow and grow and grow and grow and get water overloaded, and if they would have the potential for rupture, okay? So this is simple diffusion, okay? This is simple diffusion, except we've added a membrane, but we have solid on one side and water on one side. We have solute and water. The membrane is permeable both to solid, both to the solute, which is the red particles, and to water, okay? The membrane is permeable to both. So we can have water move on both sides. We can have solute move on both sides. So we introduce this experiment here. We've got this side with this much water in it, and this side with an equal amount of water. We got 4 solute particles on this side. We got 3 or 4 times that on the other side, and we have and we pull down. We slide a filter in there, and the filter is permeable to both. So what's going to happen after a period of time? We're still going to have the same amount of water, but the solute particles are going to go to where, okay, because this membrane is permeable to the solute particles, they're going to move and eventually at the finish line, and when equilibrium is reached, there's going to be the same amount of solute particles on each side. Okay, so that is if the semipermeable membrane was permeable both to the solute and to water, this is what had happened. Okay. But this is what usually happens now, okay? We have, on this side, we have 5 solute particles, and 10 or 15 on this side, we have an equal amount of water. The only thing that can move is the water, okay? The only thing that can move is the water. So the water moves from an area of low concentration of solute particles to an area of high concentration of solute particles through a semipermeable membrane until equilibrium is reached. So if we took a sample of a cubic millimeter, also called a cubic centimeter, also called a milliliter, they're equal, okay? 1 cubic centimeter is equal to 1 milliliter of fluid, if we're talking about water. If we took a sample of that, we would have the same amount of solute particles that if we took a sample of here, okay? Because we have more solute particles on this side, but we also have more water. We have at least twice, 2.5 times as much water, okay? So this is what osmosis does, okay? This is what osmosis does. It tries to move from an area of low solute particles to an area of high solute particles. And so these 2 slides are a good example for osmosis, and you can define it however you want to, but this is still passive diffusion. This is still a passive transport mechanism. There's no energy involved, okay? And so that's that slide. Osmolarity is the concentration of solute, okay? And so we have a specific osmolarity for our body tissues, for our blood, and you can measure blood osmolarity, okay? Tonicity. We're going to talk about 3 solutions, okay? Isotonic solution for the body is 0.9% sodium chloride. So if you see an IV that says normal saline, that's what it is, 0.9% sodium chloride, okay? So we've got a red blood cell that inside the red blood cell is 0.9% sodium chloride. And we're going to put it in a solution. We're going to put it in a solution. It's probably 0.5% sodium chloride or 0.25% sodium chloride. So we have less solutes in the solution. So we're putting this cell into a hypotonic solution. When we do that, assuming water can go in, water's going to go in, and the cell's going to get bigger, and the cell's going to get bigger, and the cell's going to get bigger. And eventually the red blood cell can lyse, okay? The red blood cell can lyse because of that movement of, we placed a red blood cell in a hypotonic solution. Hypotonic means less solute per milliliter. Hypertonic, we're doing just the opposite now. We've got a red blood cell, which inside is 0.9% sodium chloride, which is normal saline. We're now putting it in a 2% or 5% normal saline solution. Well, what's going to happen now? There's more water inside and less solute inside the red blood cell because it's 0.9%. Outside, it's 5% normal saline. So the water is going to go out of the red blood cell and it's going to shrink, and this is called crenation. And then finally, if we take that red blood cell, which is 0.9% normal saline, and we put it in an IV solution of normal saline, which is 0.9% normal saline, we're going to have water going back and forth, but the net movement of water is 0. The net movement of water is 0. Water's going to go out, water's going to go in, because that's what happens in reactions. There's constant movement. It's just that it doesn't just sit still, but the net movement would be 0. So those are 3 things that could show up on a matching, a multiple choice, a true, false. And so you really need to have a handle on that because I can change one word on the true, false, and what was normally true is now false, okay? And so if you don't have a handle, it's going to be as simple as that. It's not going to be very complicated, okay? This is not medical school. This is not dental school. This is not nursing school where it's got to be exact. This is a survey course and a survey chapter on cell biology. And so that's what we're doing. So it's going to be as simple as what we're talking about. General name for tonicity problems, water goes where the stuff is, a general rule for solving them. So if you got a tonicity problem, where's the water going to go? If it's hypotonic, it's going to go in the cell. If it's hypertonic, it's going to go out of the cell. If it's isotonic, there's going to be no net movement in or out of the cell, okay? So this is that saying, this could very well be Dr. Oler's saying, water goes where the stuff is, okay? Filtration is the last passive movement. This occurs primarily in the kidneys. You probably didn't realize it, but when fluid gets to the kidneys, when fluid and proteins and glucose, except for the cells and the really big molecules, everything gets filtered out into the filtrate. We don't call it urine until it gets out of the kidney, okay? It gets filtered out literally into the filtrate, okay? And all that's left are the red blood, are the cells and the big proteins and water surrounding them. All the rest of the stuff has been filtered out. But immediately, the fluid and the glucose and the electrolytes get resorbed. Immediately they get resorbed, okay? But this is filtration. When you put a pressure head into a compartment that has a bunch of leaky cells, and that's what happens in the glomerulus, you have a bunch of leaky cells, it leaks out, okay, into the capsule. And you know what a pressure head is. You got too much pressure and you got a lot of holes, you'll spring a leak. Well, imagine if you already had the leaks, okay? So if you got pressure coming into there, you get filtration, and that's just due to the pressure. That is not, it doesn't require energy. And that's filtration, occurs primarily in the kidney, okay? And you'll talk about that when you get to anatomy and physiology too. So all those are passive transport. This is active transport, okay? Passive transport to me is you've got the car on the hill. All you got is, literally, if it let the emergency brake off and the car is going to go down the hill. It's going to go down the hill to get to a flat space. And once the energy is dissipated, it's going to stop. Okay. That is passive. That to me is passive transport. Active transport is you had a Volkswagen Beetle. Okay. You took it wouldn't crank. You got it to the hill. You then took the brake off. You jumped in and you tried to turn in, let go of the clutch and you tried to turn the engine over, get it to start down to the bottom of the hill. But guess what? It didn't start. So now you got your Volkswagen Beetle down to the bottom of the hill, and you got to get it back up to your house because you got to call AAA to come and get it and take it to the Volkswagen place or wherever you're going to get it repaired. You're going to have you and 3 or 4 friends, okay, half the class, maybe the whole class, depending on how big of the hill, push that car back up the hill. That's active transport because you've got to put energy in, okay? Active transport is the reactions we saw, the ones that require considerable activation energy. It's when we do contractions with muscles, okay? When we cause a muscle fiber to shrink, we need energy, that's ATP. When we move ions for whatever reason against a concentration gradient, okay? Say we've got a lot of potassium inside the cell, okay? A lot of potassium inside the cell, but we want to put more because we're going to prime a pump, okay? And so we want to put more in there. Well, we can't depend on diffusion, facilitate diffusion, osmosis, or filtration to get it in there because none of those work against a concentration gradient. None of those working at concentration gradient. So we've got to we've got to have energy for that system. So that's an instance of active transport, okay? ATP, so we got to transport protein. We're going to have to, remember when we looked at adenosine triphosphate, and in that last phosphate group in that bond, there was a great amount of energy. We have phosphorylated the transport protein. In other words, we have passed that energy onto the transport protein. The protein is going to change the configuration, and some type of solute is pumped into the cell by way of the sodium-potassium pump, okay? Some type of solute is pumped into the cell by way of the sodium-potassium pump. And this is just an example of this. Secondary transport, okay? You've got active transport going on, okay? And you got hanger-on molecules. It may be, if you're trying to transport potassium potassium's an ion. An ion is an element or group of elements which has gained or lost electrons, and by way of gaining and losing electrons, has developed a charge. We went over that in chemistry part 2. Well, if we've got trying to transport potassium in, okay, we may have a tag along. We might have a phosphate group, which is PO4 2-, or we might have a chloride, which is a 1-, okay, go along with it. It didn't get transported. It just went along with it because it had electromagnetic radiation. Electromagnetic radiation and electromagnetic attraction, okay? Or we could have a tag-along molecule like glucose here, okay? We're transporting sodium, okay, by way of phosphorylating the transport protein. We're transporting sodium in, and when we transport sodium in, okay, we get the glucose comes along with it, okay? We get transport of sodium and the glucose comes along with it. So that's a secondary, okay? Primary is the transport of whatever the solute. Secondary is the tag-along molecule. And then the last method of transporting stuff into the cell actually requires the cell to take in the thing. In other words, the cell will form a wall around whatever's trying to transport in and bring it into the cell, okay? So you have very large macromolecules, okay? Or you have very large particles that are going to go into the cell. The cell literally forms a pocket around it. So it forms a pocket around it and going to bring the material in, okay? Perfect example would be if you had a macrophage, okay? You have a macrophage that has come upon a bacteria, looks at the bacteria doesn't have any self-antigens, okay? So it is non-self. Bacteria is non-self. So the macrophage is going to engulf that bacteria, break it down, okay, and into component parts. Anything of it can use, otherwise it's going to break it down, kill it, and extrude it, okay? And so that's one example. If you have macromolecules, it's the same thing. You bring in macromolecules, you've got to break the macromolecules down, okay? Usually you use a thing called lysosomes, okay, which contain enzymes in it to break the molecules down. And then you utilize what you need to utilize. And the remaining portion of it, the stuff that's non-utilized, the waste, is then, so we fused a membrane around it here to bring it into the cell. We broke it down. And then we brought it back to the cell membranes, transported back to the cell membrane. It fuses with the cell membrane and then opens up its contents, okay? Opens up its contents. So this is endocytosis, and this is exocytosis. Endo means going inside. Exo means going out the door, exit, okay? Endoscope, they put a scope in your GI system, or they can use scopes anywhere and look at things. And exocytosis means going out, okay? The thyroid gland doesn't have any direct connections with blood vessels, right? Doesn't have any ducts, doesn't have any direct connections to anything, but it makes thyroid hormone. So the thyroid gland makes thyroid hormone. Thyroid hormone, this is a little bit different, but going to make some thyroid hormone. It's going to be transported to the edge of the cell, okay? The thyroid hormone is going to be the vesicle, okay, which is transporting it. It's going to fuse with the cell membrane, release the thyroid hormone into the interstitial fluid. Well, guess what's right out here, right just microns away from the cell, is a blood vessel. So it's going to go interstitial fluid, then it's going to go into the blood vessel by simple diffusion probably, okay, because the concentration of thyroid hormone in the interstitial fluid, since the thyroid gland just released it, it's going to be higher than it is inside the blood vessel. So it's going to go in the blood vessel, then taken throughout the body wherever it needs to be used. Penocytosis is when you're taking a gulp of water or a gulp of some kind of fluid and you just bring it in. So that's just a special type of endocytosis. Phagocytosis is what we talked about, where you engulf really large particles, okay? And you're going to put this membrane around it and you're going to digest it, okay? And what you're also going to do, and you don't need to know this, you're going to break it down and you're going to take its antigens, its surface antigens, and present them to another type of cell that's going to make antibodies against it. And you're going to two types of cells, one that does cellular immunity and one that does humoral immunity, which is antibodies, okay? And so that's one of the things you do with foreign material is put its antigens up on the side of your surface. Exocytosis, ejection of waste, okay? Hormones, we just talked about that. The neurotransmitters, when we get to muscle contraction, acetylcholines are released from the end of the motor end plate, the nerve, okay? And there's a gap there. There's a gap between the nerve doesn't touch the muscle. There's a gap in that gap. So how do you get that neurotransmitter off? You take it to vesicles, fuse it with the plasma membrane, okay? And it goes into that space and diffuses across the muscle. Vesicular transport, receptor mediated, used to get a particular molecule in. So if you need a particular molecule, you've got a receptor out here saying, hey, I need you, the green molecule, okay? Green M&M, we need for you to come in because we're in short supply of green molecules, and it will specifically take these in. Okay, so that is membrane transport. Now we're going to look at the specific organelles, and it's not going to be any extensive coverage of them, which is, we're just globally talking about them. So the organelles, these are the organelles, we'll look at them, okay? This is the mitochondria. Mitochondria is called the powerhouse of the cell. It makes the majority of the body ATP. It requires oxygen. ATP can be made in the cytoplasm anaerobically, okay, if you're in oxygen debt, but it only makes a couple. If you have oxygen present, you don't have an oxygen debt, then the breakdown products of that glucose molecule can go inside the mitochondria, and you can complete the cellular respiration and get between another 30 and 36 ATP. So it's much more efficient for the mitochondria to form ATP from the glucose molecule. That's called cellular respiration. Mitochondria is double membrane, an outer single membrane, and an inner membrane that's folded on itself. When it's folded on itself, that folding is called cristae. That increases the surface area for cellular respiration to go on. And the space here between, the fluid field space here is called the matrix. Okay, this is where the matrix is. This is where the enzymes are. Double membrane organelle, intermembrane highly folded, cristae to increase surface area. Outer membrane is smooth and featureless. It contains its own DNA and RNA and is able to reproduce itself. Okay. DNA generally is only found in the nucleus. RNA is found in the nucleus and outside the nucleus. But DNA generally is only found in the nucleus. But this is a particular instance where it's found in the mitochondria. So that's a got your question, okay? Where is DNA found? Generally, it's only, is there any place that DNA is found outside the nucleus? The answer is the mitochondria, okay? Or the DNA is found in all the following places except, okay? And you could have a question like that. RNA is found in the nucleus and outside the nucleus. Powerhouse of the cell, site of cellular respiration, glucose converted into ATP. This is the ribosome. It consists of a couple of units. That's all you need to know. It consists of 2 units. Their function is protein synthesis. Their location, they either can be bound to the endoplasmic reticulum. We'll look at that in just a second. They make proteins that are going to be used in the cell membrane or exported from the cell. An example would be thyroid hormone or neurotransmitter, okay? Or they can be free floating in the cytoplasm, and they're going to make proteins for use inside the cell. So they can either be bound to endoplasmic reticulum or they can be free. This is endoplasmic reticulum. Endoplasmic reticulum, this is the blue structure here, is an extension of the nuclear membrane. And I compare this to a factory wall, okay? I compare this to a assembly line, a factory assembly line, okay, where proteins are going to be made, okay? Remember we talked about proteins and amino acids, okay? Well, these amino acids are going to be put together in here, okay? They're going to be put together in here that are going to be used in the membrane and transported outside the cell. This is rough endoplasmic reticulum. We're going to look at a picture of it in a minute. This right here is smooth endoplasmic reticulum. The difference between rough ER and smooth ER. Smooth ER is that rough ER has ribosomes attached to it. So I say that rough ER has factory workers, and smooth ER doesn't have any factory workers. It's all robotics, okay? It's all robotics. So this is endoplasmic reticulum, and if the ribosomes are attached to it, it's called rough. And if there's no ribosomes attached to this endoplasmic reticulum, it's called smooth, okay? So this is rough endoplasmic reticulum. Okay, this is rough. You can see this electron micrograph. So these ribosomes are very small, and that's smooth endoplasmic reticulum. And they're a continuation of the nuclear envelope. Endoplasmic reticulum, interconnected tubes. Rough has ribosomes, smooth doesn't. Function rough, site for protein production. Smooth, in addition to proteins that stay inside the cell, site for lipid production. Calcium storage in muscle cells. So when we get to chapters 9 and 10, we'll talk about that, specifically 9. Detoxifies drugs in liver cells and synthesizes steroids, okay? So the difference between rough is just going to make protein. Smooth has some other functions, okay? Has some other functions besides making proteins for inside the cell, okay? And these are the list of functions. Okay, so that was endoplasmic reticulum. We talked about the mitochondria. Golgi apparatus, that was this next structure here. So we now have made the protein, okay? We made the protein. But the protein has some sequences that tell it to start, tell the production to start, some sequences tell the production to stop, some nonsense sequences, okay? It's not folded, it's not put together, it's not packaged, okay? That's what the Golgi apparatus is doing. So here is my factory, okay? Here's my factory. Now here is my detailing or my UPS store, whatever you want to call it. That's Golgi apparatus, okay? That's Golgi apparatus. That's the Golgi apparatus here. That's a continuation of the endoplasmic reticulum. The Golgi apparatus is eventually going to form little vesicles. Guess what these vesicles are going to do? That's going to be your UPS trucks that transport it to the plasma membrane. So it can fuse with the plasma membrane and undergo exocytosis. So you can imagine a thyroid hormone, a neurotransmitter, something that needs to get to some other part of the body being produced by that cell. And the mechanism for that is to produce it, package it, put it inside these little vesicles, which are transport, take it to the plasma membrane, fuse it with the plasma membrane. It dumps its contents out into the interstitial fluid, and right next to the cell is a blood vessel. So the contents then go from the interstitial fluid, that thyroid hormone, that goes from the interstitial fluid into the blood vessel. Stacked membranous sacs that pinch off to form vesicles, processing, packaging, transporting products, may activate proteins if necessary. Probably some of the folding goes on here. Remember we talked about primary, secondary, tertiary, and quaternary structures of proteins. Probably some of the secondary and tertiary folding goes on in these Golgi apparatus, okay? That was the Golgi apparatus there. See, I told you we were going to talk extensively about these. Lysosomes. Lysosomes are those packages of degradatory enzymes that attach to foreign material or attach to old, worn-out organelles to break them down, digest particles brought in, digest old, worn-out, unneeded organelles, and break down needed substances, okay, or storage sacs of hormones. They break down things as they come in or as they are processed and digest them, and we utilize what we can, and everything else gets extruded. And that's a lysosome, okay? Has a lot of very powerful enzymes. They're very small, obviously, but it has a lot of very powerful enzymes, okay? Break down non-needed tissue. The webbing between the fingers is broken down by the lysosomes. As we mature as a fetus before delivery, we have webbing. The webbing's broken down. Also, break down a bone into component parts for the calcium. We have peroxisomes, which is a specialized type of a vesicle which contains hydrogen peroxide, okay? This is hydrogen peroxide. H2O2 is hydrogen peroxide. H2O is water, okay? Neutralizes free radicals, detoxes alcohol and drugs. And there's a picture of a peroxisome somewhere. I'm not going to give you this slide and ask you to pick out things. It's just too busy. What you're going to need to know is the information about it. We have a cytoskeleton in the cytoplasm to give the cell support, okay? Otherwise, it'd just be a bunch of organelle and goos, okay? A bunch of organelle and cytoplasmic gel, okay? Give support to the cell and aids in movement. Some cells move, okay? We have myofilaments, primarily actin. There's actin in most all cells, and particularly in the muscle cell, you have actin and myosin, and their interaction causes the muscle cell to contract. That's all a muscle cell does is contract, okay? That's all it does is contract. It doesn't do any high neurological movements. It just contracts, and when it contracts, if enough of them contract, you get movement, okay? So it also can use this if you have endo or exocytosis. You're going to have, number one, you're going to have something that has the capability of moving, okay? And you're going to have energy. Remember, endocytosis and exocytosis are parts of mechanisms that require ATP, their active transport. In motility of the cell, with myosin, use to brace the cell membrane, give it shape. We have these bigger molecules. Okay, these are still myofilament molecules, but they're bigger molecules that add some structure. We'll look at keratin when we do our tissue lecture. We have microtubules, okay? These compose the cilia and the flagella, okay? Some cells have cilia, okay? Some cells have flagella. Sperm cells have flagella so that it can move, okay? And so they're composed of tubulin. This is just bigger. This is an extension onto the cytoskeleton, but this is just bigger protein tubules, okay? We have the centrioles, which are responsible for tubular organization. Okay, they're the attachment point for cilia or flagella, or during cellular division, which we'll talk about on Wednesday. They migrate to the poles. They spin out spindle fibers, and those spindle fibers will attach to the chromosomes. So microtubules are 9 triplets, and they're at right angles to each other. Basal bodies for the cilia and flagella. Organized spindle apparatus when separating chromosomes during cellular division, paired at right angles. That's all you need to know about the centrioles. We have microvilli here. Okay, these are microvilli. We have some cilia. We have cilia in our respiratory tract that are constantly beating towards the mouth. Any very small particles, not a piece of popcorn, but a small particle that you inhale, they'll be trapped in this mucus, and these cilia will move it towards the mouth so you can swallow it rather than going down into the respiratory tract. So these are some cell extensions. Okay. There was the centrioles. These are the centrioles there, microtubules. The nucleus is one of the largest organelles. One per cell, except for so there's generally one nucleus per cell, except for skeletal muscle, which is voluntary muscle. It's the muscle that moves us. Some liver cells, the bone destruction cells, and we'll find out the name when we get to 6, 7, and 8. Okay. And mature red blood cells have no nucleus. They jettison their nucleus and most of their organelles before they mature, so that they are equipped to carry the most amount of hemoglobin. Okay. They jettison their nucleus. So obviously they can only make a few ATP because they don't have any mitochondria. They've jettisoned those. Okay. And most of their organelles, so they have to go by cellular respiration. They have to do the first part of cellular respiration without oxygen. And so they don't make very many ATP, but they don't need very many ATP. Okay. Contains instruction for all body proteins larger than other organelles. 3 distinct regions. Okay, you have the nuclear membrane, which is a double membrane. Outside is continuous. Remember, everything's sort of continuous. Nuclear membrane is continuous into the plasmic reticulum. And the plasmic reticulum is continuous to the Golgi apparatus. Golgi apparatus is going to break off into vesicles. Vesicles is going to fuse with the plasma membrane. Okay. It contains pores, okay, to make it selectively permeable, because if we need to get a signal into the nucleus, saying, hey, we need more thyroid hormone, open up your DNA and give us the blueprint for making thyroid hormone. Because remember, the nucleus contains the DNA for everything in the body. Turns a blueprint for every cell, every protein in the body is in the nucleus, okay? We have nucleoli. They're dark staining. They are where the ribosomes are made, not membrane bound, can be multiple in each nucleus. Okay. We have the genetic material, which is wrapped up like this. This is the DNA. It then has histone proteins that wrap it up, and eventually it forms chromosomes. Okay. So the chromatin, which is the DNA, okay, wrapped up in histoproteins, is composed of DNA and histoproteins. And this is what DNA looks like if you get a cell that's dividing, and you can see the individual chromosomes. And so that's the nucleus, and that's the end of the cell. So like I said, it's just a brief survey. It's just a brief survey of the cell, what goes on with the cell. So let's look at, go back here. Cytology PowerPoints, modules. And so we'll go down to the module or chapter 3. We'll go down to the lab part. We got 2 activities. We got activity 3 and activity 5. It's going to give us the instructions for activity 3 and activity 5. Going to tell us how to get there. Okay. We go to launch study area, select textbook chapter, select cells. Okay, that's the chapter we're in. We go to 9.1, and then we got to click on 10, and we go to exercise 1. We do activity 3. Okay. Then once we get it done, This is the complicated part, and this is why I made the video. And I'll show you where I made the mistake at on the instructions. You go through these instructions, you just click, 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 click. You put your name in, okay? Then you get down to this stage where you have the dialog box. The instruction I left off, okay? You have to click Microsoft Print to PDF and click Print to File. You click Microsoft Print to PDF and Print to File. And it's in the video, and then I'll change the instructions. But I didn't get a chance to do that, but I did get a chance to do the video, okay? And so click Print to File. The one thing I didn't write is click on Microsoft PDF, because if you anything else here won't come up as a PDF file. But if you click on those two things, and this was wrong because it didn't come out as a PDF file, but you're going to name it last name, first name. You're going to call it Cell Lab Activity 3, Cell Lab Activity 3, and PEX just means Physio X, okay? So when we get to look at it, okay, simulate an osmotic pressure, so you click on it, okay? What's going to happen first is it's going to tell you the objectives, all right? This is your objectives. It's going to be composed of several parts. It's going to tell you the objectives. It's going to give you some information to read, okay? It's going to give you some information to read, a couple of slides to read. And talk about isotonic, hypertonic, hypotonic, okay? And then it's going to give pre-lab questions, okay? Do your best on the pre-lab questions. Do I think you're going to get them all right? No, okay? They're pre-lab questions. You haven't even done the lab yet. They're asking you general things about the lab. But you should try your best, okay? You should try your best. The whole thing about the lab is you make a good faith effort. A red flag to me is, number one, you did the lab and all your experiments come out with the same results. Number one, that's you didn't do the lab. Number two, you have post-lab questions, okay? You leave blank. You leave pre-lab questions blank. You leave review sheet questions blank, okay? That's a red flag. You didn't do the lab, okay? And the reason you didn't do the lab is you probably waited to the last minute. To tell you the truth, that's probably why you didn't. So you're going to have some pre-lab questions. Which of the following is true of osmosis? Acquires energy? Nope. Movement is against the concentration of a gradient of water? Nope. It's a type of diffusion? Yes. Refers to the movement of solutes? Nope. Okay, so we'll check that one. We can check our answer if we want to. Got it correct. We go to the next one. Which of the following occurs when a hypertonic solution is added to cells? Hypertonic solution added to cells. We got 0.9% in the cell. We got 5% outside. Water is going to move towards the stuff, okay? Water is going to move towards the stuff. We got 0.9 in, 5% out. It's going to move towards stuff, so it's going to move out. The cell's going to burst? No. The cell's going to swell? No. Water's going to enter the cell? No. Cell's going to shrink. Check answer. Correct. Okay. The net movement of water would be into the cell in a hypotonic solution, in a hypertonic, hypotonic, isotonic, hypnotonic solution. See, they just threw you in a ringer there. Well, if you're going to have water moving into the cell, water moves where the stuff is. So the water outside the solution outside the cell has to be less than the solution inside the cell, and that's called hypotonic. And we click our answer. Okay, correct. Okay, we're going to click submit. If you get the answer wrong, you get the answer wrong. You're in the learning process. This is the pre-lab quiz. You click submit. Okay, remember you're going to have a pre-lab quiz, you're going to do the experiment, you're going to have a post-lab quiz, then you're going to have a review sheet. Pre-lab quiz will tell you if you got the answer right or wrong. Post-lab quiz will tell you if the answer got it right or wrong. If you do the experiment right and you get 4 out of 5 of the post-lab questions wrong, that's a red flag, okay? You get 1 wrong, that's not a red flag. You just guessed wrong. I can guess wrong, okay? I can guess wrong. And I have guessed wrong, okay? But now we're to the experimental part. It says drag the 20 MWCO membrane, okay? Molecular weight, CO membrane, to the membrane holder between the beakers, okay? Well, we look over here, so here's a 20 and here's a 50, okay? So we're just going to drag that there. And we're going to click next, okay? Increase the sodium concentration to the dispense left beaker by selecting the plus button, okay? And so you just select the plus button and increase the concentration. And here's the plus button here. So you just increase the concentration. All you do is follow the steps. Okay, all you do is follow the steps. Usually the first time through, and I'll give you an example. The Anatomy and Physiology 2 class, their first PhysioX experiment, was doing blood typing. Okay, first time through, they had to pull out the slide. They had to put individual drops of blood in the 3 chambers. They had to put anti-A, anti-B, anti-RH in each chamber. They had to mix them up. They had to take a slide over the light box. They had to cut the light box on. Then they got the results. They had to record the results, and then they had to pick out the results where it's positive or negative, and then they had to record the results. I haven't done this one in a while, but usually that's what you do for the first couple of times through. And then after that, they just have you to do the simple thing, then to make your decision and record your results. And you'll have a button whenever it gets ready to record results, saying record results. You won't have to type anything in, okay? You'll record your results from doing the experiment. So you just follow these steps. And it won't let you do it if you don't do the step right. So you can't mess it up, okay? You can't mess it up unless you put the wrong concentration solution there and said to go ahead, okay? But you can't mess it up. So you do that, and then you get to the post-lab quiz. And the post-lab quiz will be questions about your experiment. So it'll ask you to go back, says, do you want to see the results of your experiment? You say yes. So it takes you back to the results sheet, okay? You look, see what the result says, because it may be say an experiment 4 or whatever, in result 4, this, what was happening with this, and this, okay? And you go back and look and you put the answer down. In the post-lab quiz, if there's 5 questions, you certainly should get 4 out of 5. You certainly should not be missing 3, 4, and 5 out of the post-lab quiz, because you have done the lab and you've done the post-lab quiz. So that's a red flag. Then finally, you will have review questions. The review questions ask you to take what you learned and apply it, okay? And all I ask you to do, don't leave them blank. Make a good faith effort to answer the question. Even if you get it wrong, you've made a good faith effort, okay? If you get them all wrong, I might say, in the comments when I put the grade there, you would get something less than full thing because you got them all wrong. That means you weren't thinking about right there. What I would write down is say, hey, don't you want to go back and look at these answers and maybe change them and send them back to me so you can get full credit, okay? Whole purpose of this is to get full credit for the thing. Whole purpose of this is for you, since we don't have face-to-face labs, is to do a lab simulation and to learn something in the process. It's not busy work. You saw on the thing, there were 4 or 5 of them, okay? I could assign you all 5 of them. But two is plenty, okay? 2 is plenty. And the ones I picked out were active transport and osmosis, okay? So those are 2 of the more difficult concepts to get a grasp on, okay? And so that's your lab. Now, once you get done with it, you're going to go through the thing completely. You're going to get done with it, okay? Now, what are we going to do, okay? What are we going to do here once we get done with it? We're going to go down to modules, okay? And we're going to go down to the thing that I just put up, how to download and save a PhysioX assignment to a PDF file. So you're going to get to the end of it. It's going to say click print. You're going to click print. Then what do you do, okay? Here's what you do. This is 1.5 minutes, I think. Not very long. It's just me trying to find my, this was on chapter. Okay, this is a short video showing you how to save a PhysioX assignment to a PDF file. You've gotten this far now. You've saved to print, so print save as. You'll get this dialog box up. You need to click Microsoft Print to PDF and Print to File. When you do that, you will get this as a PDF document. See PDFs down there at the bottom. We'll save it again. I'll just change the name, Cooper Sheldon. This was in chapter 17 for the blood people. You just put it whatever the lab says to put it as. And I'll look for a folder to put it in. I'm going to look under my PC. I've got documents. I've got a folder that I said Anatomy and Physiology 2 assignments. I'm going to click on that. I am now going to save this. I have saved that to that folder. And should be done, but let's check here. And we'll go down to Documents. We'll go down to, here's Documents, Anatomy and Physiology 2 assignments. Here it is, Sheldon Cooper. And there is the assignment. So hopefully that will help you out. That's all you have to do to be able to save it. And then once you get ready to upload it, you're done. So suggestions, number one, I would make a folder. Okay, make a folder and label it whatever you want to. I have a ton of BSC folders, so that's why I didn't label it BSC. I wanted to label it something else. So I just said, for that one's Anatomy and Physiology 2. You know, you can label anatomy, you can label it whatever you want to. Okay, but I would put all my assignments in that folder, because invariably you're going to have, especially if you don't do your assignments on time, you're going to have a bunch of labs outstanding. You're going to load up the wrong assignment in the wrong place. Okay, you're going to put the wrong assignment in the wrong place. I'm